Now let's talk about glycolysis. As the name implies, glycolysis. So in this process, we are going to break down glucose into two molecules of pyruvate. So the glycolytic pathway is a pathway where we are going to break down monosaccharides into kind of 3-carbon compound known as pyruvate. So we have glucose, we have fructose, we also have galactose. But for this video, we talk about glucose metabolic pathway in glycolysis. So we also have fructose, which can be converted into its own product, then later joins the glycolytic pathway, as well as galactose can also be used. But today we'll be talking about glucose metabolism. So for glycolysis, you need to know this, that the goal is to convert glucose, which is a 6-carbon compound, into 2 molecules of pyruvate. So remember, pyruvate is a 3-carbon compound. So you already know the structure of glucose. By the way, you can use this structure or you can use the cyclic form. So this is glucose, which has to be converted into pyruvate. So this is a 6-carbon compound, 1, 2, 3, 4, 5, 6, and a 3-carbon compound, 1, 2, 3, which is pyruvate. So there are 2 molecules of pyruvate that are going to be formed. But the actual goal is to provide energy, and the energy is in the form of adenosine triphosphate. It's also very important to note that glycolytic pathway glycolysis is regulated, and as you already know that insulin is the most important hormone which activates glycolysis. How does it activate it? We'll talk about them when we talk about each steps of the glycolytic pathway. And it's also very important to note glycolysis has, most scholars divide it into 2 parts. So we have where we are going to spend, and there's one where we are going to gain. So one which is the preparatory phase. In the preparatory phase, we are going to use ATP. 2 molecules of ATP are going to be used here in this process. So it's like working in order to get paid and using the money to buy food to eat. So when you work, what do? You are going to use energy, right? Good. But at the end of the day, when you are going to be paid, you are going to use your money to buy food. And the food you are going to use to buy, the money you are going to use to buy the food is going to give you energy at the end of the day. But at the end of the day, what is the most important thing is profit. So the net gain of energy. The net gain of energy is the most important. So even though in glycolysis we are going to use 2 molecules of ATP, but we are going to gain more ATP than we use. So also in human organism, profit is the most important. So let's start with the reactions of glycolysis. So also very important to note that for you to understand any carbohydrate metabolic pathway, you have to understand how to draw these structures, or at least understand how to, you have to know how to recognize them. As well as you need to know the enzymes that are involved, their classes, as well as the products. And also, if possible, their regulation. So the first reaction in glycolysis is the conversion of glucose into glucose 6-phosphate. So we have glucose, which is a 6-carbon compound, glucose is converted into glucose 6-phosphate. So the first reaction is the conversion of glucose to glucose 6-phosphate. But how are we going to do this? As you can see, what's the difference? The difference between these two structures is the phosphate, right? So on glucose, you have no phosphate on the 6th carbon. So you have 1, 2, 3, 4, 5, 6. So what we're going to do is to add phosphate to glucose. So this process is known as phosphorylation. So glucose has to be phosphorylated into glucose 6-phosphate. But where are we going to get the phosphate from? We'll get it from ATP, adenosine triphosphate. And where is the rest going? It's going to become adenosine diphosphate, ADP. Meaning we have lost a phosphate group from ATP to become diphosphate. But what enzyme catalyzes this reaction? There are, well, two important enzymes you need to know. We have hexokinase and we have glucokinase. So let's write it here, hexokinase. So it's very important to note that hexokinase and glucokinase are catalyzing the same chemical reaction, the conversion of glucose to glucose 6-phosphate, but they differ in their tissue specificity as well as their activation and inhibition processes. Hexokinase, as the name implies, hexo means 6 carbon, right? For example, hexose, like glucose, mannose, fructose, galactose. Kinase means to add phosphate. So it belongs to the second group, well, class of enzymes known as transferases. Glucokinase means glucose and kinase. That means adding phosphate to glucose. Hexokinase means adding phosphate to a 6-carbon carbohydrate. That means glucose falls under hexoses, right? So the difference between, one of the differences between these two enzymes is that hexokinase is not just specific to glucose. It works on the hexoses, so it can be galactose, it can be fructose, it can be glucose. For glucokinase, as the name implies, it acts on only specific to glucose. Also, their tissue specificity. Glucokinase is exclusively found mainly found in the liver and the pancreatic beta cells. For hexokinase, it's found mainly in extrahepatic tissues, so it has a wide distribution. It's also very important to note that this reaction, the conversion of glucose to glucose 6-phosphate, is irreversible. So in glycolysis, there are 3 reactions that are irreversible. So it means these reactions are regulatory reactions. They are regulatory steps. That means substances which activate or inhibit glycolysis act on the enzymes that catalyze these reactions. For example, glucokinase. Well, as you already know, insulin is one of the substances which activate glycolysis. Well, one of the enzymes that it activates is glucokinase. So when you activate one of the regulated steps, that means you activate the process. Next is the conversion of glucose 6-phosphate into its isomeric form. Into its isomeric form. As you already know what an isomer is, isomers are compounds that have the same molecular formula but they differ in their structures. As you can see, these two structures have the same number of carbons, same number of oxygen, hydrogen, phosphate as well. But the difference is how they are positioned. The C-double bonded or the carbonyl group and the OH group are in different positions. For example, on the first carbon in glucose, we're having a carbonyl group. But in fructose structure, we're having the carbonyl group on the second point. So this one is fructose 6-phosphate. fructose 6-phosphate. So, what do you think about this? Since they are the same, what enzymes do you think should catalyze this reaction? Very important to know that when you convert between two isomeric forms, you use an isomer. And the name of this enzyme is known as phosphohexose isomerase. Phosphohexose isomerase. What do you need to also know about this? Well, the name, as the name implies, phosphohexose isomerase. That means it is an enzyme that converts between the isomeric forms of a hexose, that means a 6-carbon carbohydrate that has a phosphate. So that's why we call the enzyme phosphohexose isomerase. And it's very important to know that this reaction is a reversible reaction. Reversible. So we can go both either way. So we can glucose 6-phosphate can be converted into fructose 6-phosphate, as well as fructose 6-phosphate can be converted into glucose 6-phosphate using the same enzyme. The next reaction that we have is the conversion of fructose 6-phosphate. So now that we have a free point and OH at the extremity, we'll try to add a phosphate to it. So meaning we are going to have a phosphate on the first carbon So now we have a 6-carbon compound that has 2 phosphates attached to it. So this compound is known as fructose 1,6-bisphosphate. Sorry. Why do you call it that way? It's obvious, right? That the phosphate groups are on the first and on the 6th carbons. So that's why we call it fructose 1,6-bisphosphate. And the enzyme that catalyzes this reaction is called phosphofructokinase 1. It's very important to note that phosphofructokinase 1 is the enzyme that catalyzes the rate-limiting step of glycolysis. The reaction which converts fructose 6-phosphate, this compound, into fructose 1,6-bisphosphate is phosphofructokinase, and it catalyzes this rate-limiting step. What does it mean, rate-limiting step? It is kind of like the slowest reaction, and it is the most important because it serves as an on-off switch mechanism. The name implies phosphofructokinase 1 means phosphate that is attached to fructose. We are going to add another phosphate, that means kinase, to it, which is going to give us, but why do we put 1? That means on position number 1. Do we have any other one that we call phosphofructokinase 2? Yes, we do have it. Phosphofructokinase 2 is an alternative pathway which helps you to, so we have phosphofructokinase 1, then we have phosphofructokinase 2. So in this reaction, it's very important to note, it's better illustrated using a cyclic structure. So we're going to have So we're going to have phosphate attached to this. So now we have fructose 2,6-bisphosphate. Why? Because the phosphate group is attached to the second carbon and on the sixth carbon. So we have 1, 2, 3, 4, 5, and 6. Because the phosphate groups are attached on the second and on the sixth carbon, that's why I call it fructose 2,6-bisphosphate. Very important to note that fructose 2,6-bisphosphate is very important because it helps to activate glycolysis. It's also one of the substances that we use to activate glycolysis. And the next reaction that you need to know is the conversion. So now we have fructose 1,6-bisphosphate. What I'm going to do next is to split it into, but remember, there's a very important thing to note here. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate with the help of ATP. ATP is converted into ADP. It means now we have used 2 molecules of ATP. So, as you can see here, in the first reaction, reaction 1, we have used 1 molecule of ATP. And the third reaction, we have used another molecule of ATP. So these reactions require ATP. So this part of glycolysis is a preparatory phase, wherein we are going to spend ATP molecules. Now that we have fructose 1,6-bisphosphate, it has to be split into 2. So we are going to split it into 2 equal halves. So here we are going to split it. So what does that mean? We are going to take this hydrogen here and add it to this point. So we are going to have now, given that we have added the hydrogen to this carbon, now we have CH2OH plus, what is left here? We're only left with 1 hydrogen molecule, or 1 hydrogen atom. So we're going to be left with C double bonded O with 1 hydrogen. And the remaining phosphate plus the OH. So now we have 2 3-carbon compounds, which we have gotten from fructose 1,6-bisphosphate. The name of the enzyme that catalyzes this reaction is called aldolase. Aldolase. We'll also be talking about the 2 different forms of aldolase. We have aldolase A and we also have aldolase B. For aldolase B is involved in the fructose metabolic pathway, but aldolase A is the one that catalyzes this reaction. But we'll talk about fructose metabolic, chain later in another video. So these 2 compounds that have been formed, look at them keenly. So we have 1, 2, 3. 1, 2, 3. But what is common between them? Both of them are 3-carbon compounds. Both of them have OH in them. What comes to your mind when you hear 3-carbon, then OH? What comes to your mind? Glycerol, right? So glycerol is a 3-carbon compound. It's a triol, right? So this is glycerol. Glycerol has 3 carbons and 3 OH groups. Why is it important to understand the structure of glycerol and its name? Because we're using, the backbone is used in a lot of compounds. especially lipids. And also here, what are we having here? In this compound here, it looks like having 1, 2, 3. It looks like glycerol, but it also having a C double bonded O at the end, which makes it an aldehyde. And it also has a phosphate. So when we want to name it, we'll say glycere, from the word glycerol, because it's having its O, C double bonded O at the end, we say aldehyde, glyceraldehyde. And because the phosphate is attached on the third carbon, we say glyceraldehyde 3-phosphate. Very important to understand how to name this compound. And the other compound that is formed from this fructose 1,6-bisphosphate is called dihydroxyacetone phosphate. Why do you also need to understand another structure known as acetone? This is the structure of acetone, right? Let's say we add 2 OH groups to acetone. OH here, then another OH. It becomes dihydroxyacetone, right? Dihydroxyacetone, right? And after adding OH to acetone to form dihydroxyacetone, now let's add a phosphate group to it. So a phosphate group, let's represent it by this, phosphate. Now instead of dihydroxyacetone, now it has become dihydroxyacetone phosphate. Right? So, why is it important to note this? Because this structure and this structure are isomers. You need to know that they are having the same number of carbons, the same number of hydrogens, the same number of phosphate. So, what do you need to know? That the difference is the C double bonded O for dihydroxyacetone phosphate, DA, let's represent by DAP, dihydroxyacetone phosphate. The C double bonded O is on the second carbon. And for glyceraldehyde, GAP, 3-phosphate, the C double bonded O is on the first carbon. Next reaction that we have to do, and remember, this reaction is a reversible reaction. That means we can go either way, both ways. Next reaction is the conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. That is, so we have this converted into this. Dihydroxyacetone phosphate, like I said, and glyceraldehyde 3-phosphate are isomers. So we have to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. You can guess the enzyme. It is an isomerase. Just like the conversion of glucose 6-phosphate into fructose 6-phosphate, we use an isomerase known as phosphohexose isomerase. Here we are going to use what's called triose phosphate isomerase. So we're using an isomerase enzyme. Very important to note that this reaction is also reversible. Now, this is very critical. Why? Because when we are going to calculate how many ATP molecules we are going to get from glycolysis, we'll be having some doubling. Where is the doubling going to come from? The multiplied by 2. It's coming from here. Remember, from fructose 1,6-bisphosphate, we have gotten 1 molecule of dihydroxyacetone phosphate and 1 molecule of glyceraldehyde 3-phosphate. Now we are going to convert the dihydroxyacetone phosphate that we got from fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate. That means we'll be having 2 molecules of glyceraldehyde 3-phosphate. And please keep this in mind because we'll be using it in our subsequent calculations. Now that we have 2 molecules of glyceraldehyde 3-phosphate, right? 2 molecules of glyceraldehyde 3-phosphate. So remember, glyceraldehyde, that means we're having from the word glycerol, 3 carbons, aldehyde, that means C double bonded O, right? 3-phosphate, that means on the 3rd carbon, we're going to have a phosphate group attached. Then on the center carbon, we have an OH. So we have glyceraldehyde 3-phosphate, right? So we are going to have 2 molecules of this. Glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is going to undergo a reaction to convert it into an acid. Meaning, well, we're going to add another phosphate to it in order to have 2 phosphates, right? So we're having this and we're having this with the phosphate. So we're having this compound, but where are we going to get this phosphate from? Remember, mostly from the other reactions that we did, most of the phosphates were coming from ATP, right? But in this reaction, the conversion of glyceraldehyde 3-phosphate into this, which is 1,3-bisphosphoglycerate. Let me write it here, 1,3-bisphosphoglycerate. We're going to get the phosphate from inorganic phosphate. That's represented by Pi. So we're not going to get it from ATP. It's not ATP that we're using in this process. We're using inorganic phosphate. So you already know the structure of phosphates like this, H3PO4, right? So we're going to, what's going to happen here? We're going to remove hydrogen. So as you can see, this hydrogen from here and the hydrogen from here are going to be removed and they are going to be added to NAD+. That's nicotinamide adenine dinucleotide into a reduced form of nicotinamide adenine dinucleotide. So, and an extra hydrogen, a proton, right? So, what does this mean? We're removing 1 hydrogen from glyceraldehyde 3-phosphate and 1 hydrogen from inorganic phosphate, which will lead to the formation of NADH and a proton. But how many molecules of this are we going to have? Remember, we are using 2 molecules of glyceraldehyde 3-phosphate. You already know where we got the 2 from, right? So it means 2 molecules of NAD+ will lead to the formation of 2 molecules of NADH and 2 protons. What is the name of the enzyme? So of course, 2 things have happened here. One of the things that happened was to add a phosphate. But the most significant thing that happened is the removal of the hydrogen. Because the removal of the hydrogen has led to the formation of 2 molecules of NADH. Meaning, it's better to use the word dehydrogenase as the name of the enzyme. So we'll call the enzyme glyceraldehyde 3-phosphate dehydrogenase because we are removing hydrogen from glyceraldehyde 3-phosphate. So we'll say glyceraldehyde 3-phosphate dehydrogenase. And remember, this reaction is also a reversible reaction. Why is it important that we have formed NADH? Well, all the other steps that we did before this were preparatory reactions, because basically we used ATP. But here we are forming ATP. Very important to note that the process of glycolysis takes place in the cytoplasm, and the NADH that were formed cannot give us energy in the form of ATP without having to go into the mitochondria. So 2 molecules of NADH has to go into the mitochondria. There are pathways that we'll talk about. We'll talk about the malate aspartate pathway and glycerol 3-phosphate pathway. So, but that's for another video. The 2 NADH molecule that we have gotten is going to go into the mitochondria. There we have the electron transport chain. In the electron transport chain, what happens in the electron transport chain? Oxidation and phosphorylation. Meaning, oxidation means we are going to remove the hydrogen from the NADH proton, right? So that's oxidation. Then phosphorylation, you already know that we form ATP. There's a complex, well, known as ATP synthase, where we are going to add phosphate to ADP in order to form ATP. That's called phosphorylation. So that process by which we are going to get energy in the form of ATP from NADH using the electron transport chain is known as oxidative phosphorylation, because we are going to remove hydrogen from NADH. So due to a gradient, you already know there's an electrochemical gradient that's going to be created, and this is going to drive the work of the ATP synthase. Very important to also note that most textbooks have different, well, many different textbooks have different values of ATP that we're going to get from NADH. But it's very important to note that it depends on the shuttle. So if, for example, we are using malate aspartate shuttle in order to get the NADH into the mitochondria, we are going to get 5 ATP molecules from 2 NADH will help us to generate 5 molecules of ATP. If we are using the malate aspartate shuttle. But if we are using the other shuttle, which we call glycerol 3-phosphate shuttle, in order to get the NADH into the mitochondria, we are going to get 3 molecules, 3 ATP molecules. That's if we use the glycerol 3-phosphate shuttle. That means the most efficient of these is the malate aspartate shuttle, which provides 5 molecules of ATP. Next. Now that we have 1,3-bisphosphoglycerate with the help of glycerol 3-phosphate, glyceraldehyde 3-phosphate dehydrogenase, we have to remove 1 phosphate from this compound, from the first one. So we have a phosphate here. So this phosphate can be removed. How? So we are going to remove the phosphate that we have here on the first carbon and add it to ADP, and we're going to form ATP. What is that going to lead to? It's going to lead to the formation of three phosphoglycerate. So 3-phosphoglycerate, meaning we only have the phosphate on the third carbon. 1, 2, 3. So on the third carbon we're having a phosphate group. That's what we are going to call it, 3-phosphoglycerate. What do you think? What's the name of the enzyme that catalyzes this reaction? It's very important to note that this reaction is a reversible reaction. That means the enzyme that catalyzes it can do it either way. So we can either convert 1,3-bisphosphoglycerate into 3-phosphoglycerate, or 3-phosphoglycerate can be converted to 1,3-bisphosphoglycerate. But please note, the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate is a glycolytic reaction. But the conversion of 3-phosphoglycerate into 1,3-bisphosphoglycerate is a gluconeogenic reaction. So it's a reaction that's involved in gluconeogenesis. That means we are going backward in order to synthesize glucose finally. The enzyme that does this reaction is a kinase enzyme. Phosphoglycerate kinase. So means we have formed an ATP molecule from one molecule of 1,3-bisphosphoglycerate. But remember, we started with 2 molecules of glyceraldehyde 3-phosphate. That means we are going to form 2 molecules of 1,3-bisphosphoglycerate. Means we are going to use 2 molecules of ADP to form 2 molecules of ATP, as well as 2 molecules of 3-phosphoglycerate. The next reaction that we're going to have is the conversion of 3-phosphoglycerate into 2-phosphoglycerate. That means we are going to switch the phosphate from the 3rd carbon to the 2nd carbon. So we're having it here. Now we're going to take it, the phosphate, and attach it here on the 2nd carbon. So instead of 3-phosphoglycerate, now we'll call it 2-phosphoglycerate. What's the name of the enzyme? So given that the difference between 3-phosphoglycerate and 2-phosphoglycerate is the position of the phosphate, so expect a kinase, right? And well, we'll call it has a special name. We'll call it a mutase. So we are going to have a phosphoglycerate mutase. We are going to use a mutase enzyme here. And remember, this reaction is a reversible reaction. The next reaction we have is the conversion of 2-phosphoglycerate into phosphoenol pyruvate. How is that going to happen? We are going to remove water from 2-phosphoglycerate. Remember, there are 2 parts to water. We have H and we have OH. When you want to remove water, you need to know where you're going to remove the OH and where you're going to remove the H from the compound. So as you can see, we have a space here. We have OH here. And we also have one H here. So these 2 can be removed. But it's very important to note in chemistry, for structures, reactions, when we remove 2 different groups, or even just the same group, 2 groups from 2 different atoms, neighboring atoms that are joined together, we are going to form a double bond. So in this situation, because we are removing hydrogen from this and hydrogen from this, we are going to form a double bond between them. So we are going to have a double bond between the 2. So we are going to remove water from it. What does this mean? Well, that means a lyase, but we'll talk about the classes of enzymes in details later. But here we have removed a group to form a double bond, so it belongs to the class of lyases. And the specific name of this enzyme is called enolase. Enolase is the enzyme that catalyzes the conversion of 2-phosphoglycerate into phosphoenol pyruvate. Now, the last step in glycolysis is the conversion of phosphoenol pyruvate into pyruvate. So, how is this one gonna be? Remember, we still have one phosphate here. So this phosphate can be added to ADP in order to form ATP. So we're going to add it to ADP and we're going to form ATP. But remember, from the beginning that we started with two molecules of glyceraldehyde 3-phosphate. So we're forming two molecules of 3-phosphoglycerate, 2-phosphoglycerate. These are all in twos. We're forming two molecules of phosphoenol pyruvate. So it means we are going to use two molecules of ADP in order to synthesize two molecules of ATP in order to form pyruvate. As you can see here, two molecules of ADP added to form two molecules of ATP. Now we have a 3-carbon compound without a phosphate. That means we have removed all the phosphate that we need. Now we have formed pyruvate. But what's the name of the enzyme? It's very important to note that this reaction is irreversible. That means one of the irreversible reactions of glycolysis. So the name of the enzyme is called pyruvate. It's named after pyruvate. Pyruvate kinase. Well, even though it's like counterintuitive, it's like, well, it's like saying adding phosphate to pyruvate. But in reality, it doesn't happen that way. You cannot convert pyruvate directly into phosphoenol pyruvate. But we'll talk about it later in another video when we're going to talk about gluconeogenesis, when pyruvate will have to undergo certain reactions, more than just one. You're going to undergo in the mitochondria, then in the cytoplasm, in order to form pyruvate. But it cannot be converted directly into pyruvate. So now we have formed two molecules of ATP, right? So, and also two molecules of pyruvate. That's the goal of glycolysis. The main goal is to form ATP. And the result is going to form not just two ATP, but also two molecules of pyruvate. Because the two molecules of pyruvate are going to be used in other pathways. Pyruvate has to be converted either into lactate, which is an anaerobic process, or it can be used in glycolysis, Krebs cycle with the help of acetyl-CoA. And it's very important to note these things about glycolysis in summary. So we have discussed everything about the glycolytic pathway. Very important to note that there are two main phases. We have the preparatory phase and we have the payoff phase. In the preparatory phase, we used two molecules of ATP. And in the payoff phase, we formed ATP molecules. But there are two ways by which we form ATP. We have got substrate level phosphorylation and we have oxidative phosphorylation. One of the reactions that we have here, which is the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, involved what? NAD plus forming NADH. But this NADH is also an energy molecule, but indirectly. It first of all has to be converted into ATP for it to be usable. But how are we going to get the ATP from NADH? It has to go into the electron transport chain. So because we are going to form energy in the form of ATP using the electron transport chain, it's called oxidative phosphorylation. As the name implies, oxidative, we are going to remove hydrogen from the NADH, which is going to create a gradient electromotive force, which is going to drive the ATP synthase, which is going to add phosphate, inorganic phosphate, to ADP, which is called phosphorylation. That's what we call oxidative phosphorylation. And the other method by which we get energy in the form of ATP in glycolysis is called substrate level phosphorylation. Well, we don't have to go that far. So here, we are getting ATP from a substrate, directly from a substrate. We're not going to have to move phosphoenol pyruvate to the mitochondria to get energy. Nope. We're going to get the ATP directly from phosphoenol pyruvate by removing its phosphate group and adding it to ADP in order to form ATP. So this process is called substrate level phosphorylation. And it's also very important to note that there are 3 reactions that are irreversible. The first reaction, the third reaction, and the 10th reaction. They are irreversible. So the reaction that's catalyzed by glucokinase or hexokinase, the reaction catalyzed by phosphofructokinase 1, and the reaction catalyzed by pyruvate kinase. These reactions are irreversible reactions. So these reactions are regulatory. And the last thing that you need to know is that phosphofructokinase 1 is the rate-limiting enzyme. For the hormones, you need to know these things. We have insulin and glucagon. Insulin activates glycolysis and glucagon inhibits glycolysis. It's very important to know, but we'll explain better in the other videos, that insulin tries to convert glucose into something else, and glucagon tries to synthesize glucose. So most reactions which involve the conversion of glucose into something else are catalyzed by insulin. For example, glycolysis, it involves conversion of glucose into something else, that means to form ATP, right? Into pyruvate. So insulin is going to activate glycolysis. Remember, the pyruvate that has also been formed is going to be used later in Krebs cycle by the formation of 2 molecules of acetyl-CoA. Later, the acetyl-CoA is going to be converted into citrate by joining with oxaloacetate. So that's like a continuation, right? So insulin is also going to activate Krebs cycle. For example, citrate synthase enzyme is going to be one of the enzymes that's going to be affected by insulin. Let's go to the next pathway. For example, glucose is going to be converted into glycogen. So that is what glycogenesis. So it involves glucose being converted into something else, right? So insulin is also going to activate this process. On the contrary, gluconeogenesis involves synthesis of glucose. It involves formation of glucose. So insulin is against that. So insulin should inhibit gluconeogenesis. But glucagon, which likes glucose, that means it wants to maintain your blood sugar level. When you lack glucose, enough glucose, glucagon works. So it activates gluconeogenesis, which involves the synthesis of glucose. Also, glucagon also activates the process of breaking down of glycogen into glucose, which we call glycogenolysis. So, for example, we have glycogen phosphorylase, which is one of the enzymes in this process. So you expect what? Glucagon to activate glycogen phosphorylase. And what do you expect insulin to do? You also expect insulin to inhibit this enzyme. So that's why when you study the pathways, you need to know each enzyme that is involved in that pathway, the regulatory steps, because sometimes you may not need to know all the reactions, but at least you have to know the regulatory steps. That means the enzymes that are involved in those regulatory steps, because it is those enzymes that these hormones are going to act on in order to either activate or inhibit. Thank you very much, and see you in the next video.